My name is Robbie Embry, and I am presenting my test tube number 5 analysis. We wonder why it's important to be able to identify different microorganisms, and here are a couple different reasons why it is pretty important. We must know how to safely handle the organism, whether it's a BSL 1 or 2. We don't know, so we have to be able to identify it and make sure we proceed safely. We must be able to administer the appropriate antibiotic in case somebody is sick and we want to treat them for the right bacteria. Different organisms can help us understand life processes. We know that E. coli has been very generous in allowing us to study it. And we can identify different bacteria in the same genus, like different types of Clostridiums or other bacteria that can be found in different places. Here we have the supplied tests that I used. We used a number of different tests, and I was able to narrow down some different tests that I did not need to use. And I believe the gram stain is the obvious best choice to use as a starting point for these tests, as it allows us to differentiate between gram-negative and gram-positive bacteria. I was able to narrow it down to a gram-negative bacteria, as you could vaguely see with the pink rods in the microscope. And because of this, I was able to eliminate the catalase, oxidase, gelatin, starch, and lipid hydrolysis tests. From there, I wanted to use the hydrogen sulfide production, indole test, litmus milk test, citrate test, and urease test, because these individual tests would allow for differing results between the other gram-negative bacteria. And once again here we see the different tests conducted, not in any particular order, but I used the gram staining as my first test, followed with a slant. Slant's not really too much as an important test to help identify the bacteria, but it helps. From there we use the hydrogen sulfide production, an indole test, which is unfortunately not pictured. Citrate, litmus, and urease tests also followed. And as previously mentioned, we have the gram stain results, which ended up being a gram-negative bacteria in the shape of a rod. Here we have the hydrogen sulfide production, and as we can see, there is no black precipitate, which means no hydrogen sulfide gas was produced. Therefore, it is a negative test result. From then on, we used the litmus milk test, and it went from a deep-ish purple color to a pretty bright pink color, indicating that it is very much an acid. And there's a bit of a white curd at the bottom, so we can conclude that a reduction occurred with curd. I had also inoculated a citrate test tube, from which the color started out as a darkish green color, and after 4 days or so, the test tube remained a dark green color, indicating that the citrate test was a negative. From then on, I analyzed the urease test tube, which also sat for 4 or so days, and it remained the orange-yellow light color, which indicated a negative urease test. And last but not least, we had the indole test, which was in fact a positive reaction once the liquid turned into a pinkish red color for me. Unfortunately, I did not manage to get a picture of this test, so I had to snag one from Google to show what it looked like. But I did have a pink color at the top of my test tube. And it ultimately was my deciding factor into identifying my bacteria, as it met the requirements. And the slants just had like a white, yellow, glistening growth, not really too important into identifying my bacteria. And from here, I was able to identify my organism from test tube number 5 as E. coli. On the image here provided, you could see that the tests I starred with the results and observations recorded. And we wonder why E. coli is important to us. And we've used E. coli as a model organism in class, and many other scientific practices have used it to study. E. coli is, like I just said, heavily studied for the gut microbiome or cellular processes, and it can also help us protect our stomach from other hostile microbes. And here I just have it written out in words about why.